right here have no text to it, so you're going to have to write the text. It's just pictures, just pictures. So first of all, I want to go over the anatomy. Yes?
This is also stuff that will be tested on.
What's that?
Will we also be tested on the muscle material?
Oh, yeah.
Oh,
Oh, definitely.
great.
Yeah, definitely. You're going to be tested for everything I say. So next up will be ecology. Yeah. Which you're going to have some angles to think about. All right. But all right, yeah. Okay, so muscles, skeletal muscles. Okay. It's typically a muscle bundle of muscle fibers. The muscle fiber is the muscle cell. And usually these are multinucleate, because we're talking about skeletal muscle. They're multinucleate, because how do they form is in the early embryo, these single cells that will make up muscle, they fuse together, okay? So that's what makes them multinucleate. And the muscle fiber is just as long as the muscle. And so some of these can be very long. Okay, for example, the muscle here in your femur, right here, it will go, the cells will be from way up here all the way down to your knee. Okay, so they're long cells. And so that, and within the muscle itself, there's other things there too. There's nerves, there's blood vessels, connective tissues, and so forth. But it's filled with parallel layers of muscle cells, muscle fibers. The muscle fiber itself is made up of repeating units that are called sarcomeres. Sarcomeres. Should be up there on the slide, but I'll put it here as well. Okay, sarcomere. And, with an E. And it's, and within, well, first of all, before I get to the sarcomere, within the muscle fiber, it has parallel groups of myofibrils. Myofibrils are, and they're all in perfect register. Now, this perfect register will make sense a little bit later here. But still, the muscle fiber is filled with myofibrils. And myofibrils are made up cytoskeletal elements. Cytoskeletal elements, two different types of cytoskeletal elements that we have talked about last semester. Microfilaments. What is a microfilament?
Small filaments.
Microfilaments. What protein are they made out of? Actin. Okay, actin. And myosin is a motor protein. Okay. Now the types of, we have many different genes for many different types of myosin. Animals do. And this is one particular type of myosin that has a very long tail. Now, some of the myosins I was talking about before that carry cargo in your cells, they are like dynein and kinesins. They have two leg-like domains and also a cargo domain. Now, this type of myosin in here has also two binding spots on one end, which are homologous to the leg regions, domains of other myosin molecules, but then they also have a real long tail. Okay, so in fact, let's change this a little bit, because usually that's caught like this. All right, and so the way in which these myosin filaments, microfilaments, and the motor proteins, myosin, are put together, the actins and myosins, they form what are called thin filaments, thick filaments. The thin filament is just simply a microfilament, okay? Typical microfilament, which consists of two strands of actin molecules, polymers, that are wrapped around each other, okay? It's a solid rod. All of the motor proteins, the myosins, their tails are connected together, and we have hundreds of these myosin molecules that are connected together to form what is called the thick filament. Okay? sort of shown in this figure down here, thick filament. So this is a sarcomere, okay, from one end to the other. And what we have in the myofilament, myofibril, is sarcomer, sarcomer, sarcomer. It's a long chain of sarcomers. And all the myofibrils are so aligned that the sarcomers in one are in exact register with the sarcomers in other myofilaments. And so all these sarcomers are aligned even though they're in hundreds of different myofilaments. And this is what gives this skeletal muscle this striated appearance. Is because where the dark line is where the myosin thick filaments happen to be located because they're thicker and therefore absorb more light. And the light lines here is where mostly you have the actin filaments, the thin filaments. And so the regions that are in here, there's two bands that we usually see here. You can see two bands, a dark band and a light band. The dark band is called the A band. The dark band here is called the A band. And this is where the myosin motor proteins happen to be. Okay, we have hundreds of myosin motor proteins in this thick filament. Furthermore, their tails are connected to a protein scaffolding that is called the M line. This is made up of protein. Okay, now this sarcomer that you're seeing here on the board, although it is two-dimensional, this is a cylinder. Okay, it's a cylinder, and so you have all these myosin heads or thick filaments that are in a circle, and I'll show you that here in a moment. So this this protein scaffolding, the M line, is holding all these tails in place. And furthermore, this thin, thick filament is polarized. From the M line going to the left and the M line going to the right, the motor proteins are in opposite directions. Okay, they're in opposite directions. The I band is the light band because it contains only the actin myofilaments. Down the middle of the I band is a line, once again, a protein scaffolding that is called the Z line. Okay, so the Z line consists of proteins that are holding the myosin filaments in place. Okay, so the myosin filaments, not the myosin filaments, the actin filaments, the thin filaments, actin filaments, they're connected to the protein at the Z line, and they're oriented toward the M line. So that what we have here is that within the A band, there is an area that contains both actin filaments and myosin filaments. Okay, thick filaments, thin filaments, and they overlap, interdigitate. For each myosin filament, it is surrounded by 6 actin filaments. Okay, and all the heads, okay, these head pieces, they have binding site for actin. Okay, there's 2 heads here. Both of them have binding sites for actin. And what happens when the muscle is working is that the myosin heads, and we have hundreds of them, they will grab onto they'll attach to the actin microfilament, and the heads undergo conformational changes. The heads will grab and then pull like that. Okay, grab, pull. And when they do so, they're walking on the filaments here, microfilaments, and so the 2 Z lines move together. The sarcomer shortens, okay? And so we have hundreds to thousands of sarcomers along that myofibril, and all those myofibrils, they all shorten simultaneously, okay? And then for the muscle, and that leads to the contraction of the muscle. Okay, now let's get to the anatomy here. Once again, 2 bands that you see. I band, and down the middle of the I band is the Z line. Okay, and so this I band is actually overlapping 2 different sarcomers. Okay, part of it on the left, on the right here, belongs to, on the right side of the Z band, belongs to this right sarcomer, whereas part of the I band over on the left belongs to the left sarcomer. Okay, but the edge of the sarcomer is the Z band. Yes.
So they kind of, the sarcomers kind of just connect to each other because of
Yeah,
that.
this protein's, yeah, because here on this Z line, to the right, you have myofilaments,
Mhm.
and to the left are attached myofilaments. Okay, so they're going opposite directions.
I see.
Whereas with the M line, which is a protein, it's holding on to the tail pieces of the thick filaments, of the myosin motor proteins. And those myosin motor proteins going to the right, they work this way, whereas those to the left, they pull this way. So they're going in opposite directions. And when they contract, this whole thing will slide. In fact, I'll show it here. Good slide right here. So this is from a different book, but this is when it's completely relaxed. And so when it contracts, these myosin head pieces will hold on to the microfilament, and it pulls, it'll walk that direction. And I'll show you how it does it a little bit later, while it's this one here walks that direction. But since this is stationary, this M line keeps these motor proteins, the thick filaments, in place. This walking motion brings the two Z lines in. And so as we contract, what happens, incidentally, on each side of the M line is a place that's called the H zone. This is where just only the tail pieces of the thick filaments of the myosin motor proteins, only the tails are there, not the head pieces. Okay, that constitutes the H zone. And so as these muscle contracts, what happens is that the H zone and the I zone, they start to disappear. In fact, what we have here is that the I zone and the H zone are totally not there anymore until it relaxes. So the two Z lines of the sarcomer are being pulled in. Okay, everyone understand that? Okay, all right. Now, a little bit more. Go back to this figure. I gave you this figure as well. So this is showing the plasma membrane of a muscle cell is called it's called sarcolemma. I'm not going to use this word. I'm still going to call it just the plasma membrane. However, what is also important in this muscle fiber is the endoplasmic reticulum, the smooth endoplasmic reticulum, because it contains and sequesters calcium. Calcium plays a very big role here, okay? So now that is called, given a special name, it's called a sarcoplasmic reticulum, okay? But it is smooth ER, smooth ER. A little bit more here on the anatomy. Okay, this is just another figure. I got this from somewhere else. If you did a cross-section through a sarcomer, through a sarcomere, is at the M line, this is what you would see, okay? These are the myosin tail pieces, and we have proteins here that hold them all together. So that's what we have at the M line. If you go over here into the H zone, this calls it H band, but really the technical name is this here is the H zone, okay? H zone is part of the A band. The A band reaches from the ends of the thick filaments to the other end of the thick filaments. And we have the M line and the H zone right down the middle of the A band, okay? Now, so if we did a cross section right here, what we'd see is just the thick filaments without the head pieces. This is in the tail pieces of the myosin. We take a cross section right here through the A line, then what we see is the thick filaments surrounded by the thin filaments, okay? And there's 6 of these thin filaments for every thick filament, okay? And of course the head pieces on these thick filaments, they're going all directions so that they can grab onto any of these microfilaments. All right? And then if we take a cross section here through the I band, not at the Z line, but over here, then all you see is just thin filaments. Okay? This is how they're looking. There's another cartoon here that's showing the thick filament. The end line would be right down the middle. And these are the cross bridges, headpieces that have the binding site for actin. Okay? Now, there's other proteins here that are important too. Namely, that, okay, this is the myosin headpiece. Guess this doesn't show what I'm looking for. Go to something else here. Yes. Okay, now, when the myosin motor protein, it has a binding site, for ATP. Okay, and so what will happen is ATP binds to the binding site, and then it hydrolyzes. So we would have phosphate and ADP, and when that happens, it leads to a conformational change in the headpiece. First of all, the headpiece is out like this, binds ATP, that doesn't do anything to it, but then when it hydrolyzes, we still have ADP and phosphate attached, but then the headpiece cocks. Okay, first of all, it's like this, then it cocks like that. And also, it changes the binding site at the tip that makes it what to bind to. It now can bind to actin. Okay, myosin binding site on actin after it has hydrolyzed. Okay, so let me just put this up on the board. ATP binds, then hydrolyzes, so that we have phosphate plus ADP, and that leads to cocking of the myosin. Okay, first of all, it's like this, and then it becomes like that. Okay, and also that conformational change leads to exposing a binding site on the myosin heads that can bind to the actin. Okay, there's actin has myosin binding sites. However, the myosin can't bind because when the muscle is relaxed, those binding sites for myosin on the microfilaments are covered by a linear type of protein called tropomyosin. So the myosin binding sites are not exposed. Tropomyosin prevents the myosin from binding. Okay, it's covering up the myosin binding sites. Yes?
That's only during contraction or all the time?
No, when it's not contracting.
Oh, not.
Not contracting. Now for contraction, what happens is connected to the tropomyosin is another protein called troponin. And that's actually more than one type of troponin here, but we'll just call it troponin. Now, troponin has binding sites for calcium. Okay, so when a nerve impulse reaches a muscle, leads to depolarization of the muscle, and I'll come back to that because that's more of the story here. But that will lead to opening of calcium channels in the sarcoplasmic reticulum, the smooth ER. Okay, in the smooth ER, which is also a smooth ER, sarcoplasmic reticulum, there are two different types of proteins. There is a voltage gated calcium channel, and there is also a calcium ATPase. This is always working, the calcium ATPase. And what it does is it pumps calcium out of the cytosol into the lumen of the smooth ER. Now, when depolarization takes place on the muscle cell, that leads to the voltage-gated calcium channels opening. Okay, they open, and that will release calcium into the cytoplasm. The calcium will bind to troponin, because it has the binding site for troponin, leading to a conformational change, which pulls the tropomyosin out of the way, exposing the myosin binding sites. Then our myosin head pieces can bind. Okay, and they go through an action here. Okay, another part of the anatomy here. Since these muscle cells are pretty big, muscle fibers, the electrical signal needs to be able to reach all parts of the sarcoplasmic reticulum, all parts of the smooth ER. So periodically, the plasma membrane, up here it's calling it the sarcolemma, but that is the plasma membrane. The plasma membrane will invaginate deep into the muscle cell. And this is called T-tubule system. Okay, so all the T-tubule system is just deep invaginations of the plasma membrane into the interior of the muscle fiber. And so you have periodically all these tubes, which are the plasma membrane going deep in here where they're in contact with, very close contact with the smooth ER. Okay, so that's going to come up here in a moment as well. I'll talk about that. Let's go back to the myosin thick filaments. How did they go through their cycles? First of all, when they bind ATP, I started it up here, now I'll continue it. First of all, it has to bind ATP, then it hydrolyzes, that leads to the cocking and the grabbing, okay? Because now it binds, the myosin head is in high energy conformation, and if we have calcium in here, the cross-bridge binding sites, the myosin binding sites on the thin filaments are exposed, and we get binding, okay, of our myosin. Then the ATP, ADP are released. ADP, sorry. And the phosphate released. You already have that there. And that leads to the power stroke. So what we have here is that first of all, our myosin heads are like this. We bind ATP, they cock, then they grab, and then ATP comes off, and this will undergo conformational change. It's holding on to the thin filament and it goes this way, and then it will stay there until it binds ATP. When it binds ATP, we're back at the beginning and it releases. And then we cock again, grab, and power stroke. Bind ATP, comes off, grabs, power stroke. Now, when it releases, why does not the actin filament go back? Because we have hundreds of heads holding on to this, and they're doing their power stroke out of sequence. And so that when one myosin head releases, there's plenty of others that are holding and pulling the actin microfilament inward. So that when it grabs, and it goes through these cycles, again and again, until we have the whole sarcomere completely contracted. And it'll stay that way. That's called rigor, okay, until the calcium is removed. So, rigor means the muscle is contracted. The sarcomere is fully contracted, and we have to then, these channels, calcium channels that release the calcium, they're only open for a short period of time. They close, and our calcium ATPase pumps the calcium back out, back into the smooth ER. And as a consequence, the myosin, or excuse me, go back to the this one here. The calcium comes off of the troponin. The tropomyosin goes back into place. And now, even though our myosin heads have bound ATP, they're cocked and ready to go, they can't grab onto anything until the next nerve impulse comes. And we go through the whole system. Okay? Now, what happens at synapse? Nerve is attached, nerve ending, it branches, usually these motor neurons. They have many branches, and they are innervating a unit of the skeletal muscle, okay, a motor unit. And the neurotransmitter, once we have action potentials reach here, of course, calcium moves in, our neurotransmitter will bind, and the neurotransmitter on skeletal muscle is always acetylcholine. So this will be released from the presynaptic cell, which happens to be the neuron, and it will go to acetylcholine receptors on the postsynaptic cell, which happens to be the muscle. Acetylcholine, it'll bind to acetylcholine receptors, and it'll open both sodium and potassium channels, which leads to depolarization, okay? Because there's more sodium than potassium. So the depolarization, the action potentials on a muscle cell does not have the same shape of what I showed you with the neuron, okay? It's a little bit different type of action potential, but nonetheless, the muscle is polarized. You have positive charge on the outside, negative charge on the inside, just like all cells. We open sodium channels and we get depolarization. That leads to an action potential that is translated along the plasma membrane down the T-tubular system, okay? So we have those action potentials going there. And as we get depolarization deep inside, that leads to the opening of the calcium channels. Calcium channels then release calcium. Calcium binds. Calcium will bind, go back two more. Calcium will bind to the troponin complex, leading to a conformational change that pulls the tropomyosin away, exposing the myosin binding sites on the thin filaments. Our myosin proteins, they're already in the high energy state. They now can grab onto the microfilament, undergo the power stroke, bind ATP, let off, recock, and so forth. And so that goes through those cycles many times until these channels are shut, because they're only open for a very short period of time, and then the calcium ATPase reverse it. Now, so that's the whole anatomy and how it's working, okay, with the skeletal muscles. There are certain snake poisons, venomous snakes, the way they work, depends on the species of snake, but some of them, they are affecting the synapse. Some of these will inhibit acetylcholine esterase. Something I need to put that up there because the acetylcholine is broken down in the synapse by an important enzyme. Acetylcholine esterase breaks it down into an acetyl group and something called choline. Both of this, acetyl and choline, are retaken up by the nerve cell normally. But some snake venoms, they are inhibiting the acetylcholine esterase. Now, what's that going to do is that the acetylcholine is not broken down. So it's going to be bound to the muscle cells, it's not being broken down, and you're getting constant stimulation of the sarcoplasmic reticulum. Calcium is always there, the muscles go into rigor.
So you're paralyzed.
And you're paralyzed. Okay, you're paralyzed, you can't move. Some of these, what they do, some snake venoms is instead of that, they will block the acetylcholine receptor. Okay, and once again, with the acetylcholine receptor blocked, then even if you have acetylcholine released, you'll get no muscle contraction. You're paralyzed as well. Okay, and so both of those are things that happen. All right. Okay, so that pretty well covers it. I think I have, let me go back here. I think I had a movie. I didn't check to see if this movie is still available.
When a neuron stimulates a muscle cell, an action potential sweeps over the plasma membrane of the muscle cell. The action potential releases internal stores of calcium that flow through the muscle cell and trigger a contraction. Muscle cells have an elaborate architecture that allows them to distribute calcium ions quickly throughout the cytosol. Deep tubular invaginations of the plasma membrane, called T-tubules, crisscross the cell. When the cell is stimulated, a wave of depolarization, that is, an action potential, spreads from the synapse over the plasma membrane and via the T-tubules deep into the cell. A voltage-sensitive protein in these membranes opens a calcium release channel in the adjacent sarcoplasmic reticulum, which is the major calcium store in muscle cells, thereby releasing a burst of calcium ions all throughout the cytosol of the cell. Within a contractile bundle of a muscle cell, called a myofibril, the calcium interacts with protein filaments to trigger contraction. In each contracting unit, or sarcomere, thin actin and thick myosin filaments are juxtaposed, but cannot interact in the absence of calcium. This is because myosin binding sites on the actin filaments are all covered by a rod-shaped protein called tropomyosin. A calcium-sensitive complex, called troponin, is attached to the end of each tropomyosin molecule. When calcium floods the cell, troponin binds to it, moving tropomyosin off the myosin binding sites. Opening the myosin binding site on the actin filaments allows the myosin motors to crawl along the actin, resulting in a contraction of the muscle fiber. Calcium is then quickly returned to the sarcoplasmic reticulum by the action of a calcium pump. Without calcium, myosin releases actin, and the filaments slide back to their original positions.
Okay, that's it.
So the point of the T, like, the
Looks like there's another in there, but I think that will be enough. Okay, now what happens when you die? Okay, you go into a condition initially called rigor mortis. Now, why is that? Because your body stops making ATP. Okay, you don't have any ATP. So what is the condition of your muscles? Those muscles have contracted, okay, used up the ATP that was there, but for the myosin motor protein to release from the microfilament, it has to bind ATP. And if there's no ATP, it stays bound, and therefore the sarcomeres are all contracted, okay? And that is rigor mortis. Eventually, of course, that breaks down, eventually, because your whole body is falling apart. You know, things are breaking down, and even those motor proteins will break down eventually. Okay, any questions? Muscles, yes.
So, is the voltage gated calcium channels, are they in the plasma membrane of the smooth?
No, they're in the membrane of the smooth ER.
Smooth ER.
Yeah, that's a little bit more. I'm sort of taking a shortcut here because that their movie showed that there is a protein that's in the plasma membrane in the T-tubular system and the membrane of the sarcoplasmic reticulum smooth ER real close together, and that protein is in contact with this channel. Okay, and so it's a little bit more complex. I just call this a voltage-gated calcium channel, but really it's the other protein that is voltage-gated. And it leads to a conformational change that opens this channel because the two proteins are right next to each other. Okay? All right? Any other questions? Okay, that's going to be all of the animals. So I still have time. Since you have an exam tomorrow, I'll probably let you out early. But I want to get started on just the next lecture.